Okay, next we're going to hear from Quan Zhong, who's going to tell us about the functional characterization of human gene alleles using interspecies genetic approaches.
Hi, everyone. This is actually the first time I'm participating in the yeast genetics meeting. It's absolutely wonderful experience. Thanks very much for having me. To be able to present here means a lot. And oops. Apologize, I am clicking. Let me try this. Oh, okay. I'm from Wright State University, and probably a lot of you haven't heard about it at all. Me neither, until both me and my husband took a faculty job position there. And so we're situated in a city called Dayton, and that's in the southwest of Ohio. We're just like 3-4 hours away from Chicago, Detroit, and Ann Arbor, Philadelphia, and 1 hour away actually from Ohio State. And we have been quite happy since we joined the school, and I started in my lab in 2014. And in the next few minutes, I would like to introduce to some of the ideas that we're exploring at the moment. So when I first started my postdoc with Dr. Mark Vidal at the Dana-Farber Cancer Institute, and he posed me a question, and basically asking to what extent that you think the human Mendelian disease-associated mutation, these are germline mutations, they're causal, could be still functional. Functional meaning that it cannot be modeled by a simple, completely loss of function like a null allele. And at that time, I came from a yeast genetics lab, and basically all what I did is genetic screens, suppressors, and experimental work. And to answer these questions, it's quite overwhelming. But over the time, I learned to talk to computational biologists and to learn, to come up with the idea. And in about 2 years, we come up with a few things, and then we're able to, by quantifying the distribution of different types of disease mutations in dominant versus recessive disease. And we came up with the conclusion that at least a substantial of such germline mutations could still be functional. And they're leading to disease, not necessarily that the gene is gone, but rather that the function has been changed. And different mutations could potentially lead to different types of perturbation of gene functions, and therefore give rise to different phenotypes. And following that story, we developed experimental pipeline where that we can generate large-scale single-point mutations, and we use high-throughput functional assays such as protein-protein interactions to characterize to what extent the mutant alleles have changed their functionality. And despite all the excitement of this work, and we know deep down that, the complex genotype-phenotype relationships that we're trying to explain here is far from what's out there. And, the phenotype expression is very complex. And not only that different type mutation could potentially lead to different disease, in the same exact allele could be leading very different phenotypes in different individuals. And these has been classified, for example, in complete penetrance or variable expressivity. And the reason that these kind of phenomena occur, in addition to potential environmental influence, of course, there are genetic modifiers, meaning that there are other genes could potentially influence the to what extent that the phenotype of the allele X, the mutations, could be expressed in specific individuals. So we think that this is a much bigger challenge in trying for understanding the disease mechanisms, as well as for predicting potential functional and phenotypic consequences of human genetic variations. And to solve these challenges, and we turn to our favorite model organism, yeast. And yeast has been historically chosen as a model to characterize human gene alleles and to what extent that they could be functional or not functional. Here I borrowed a beautiful figure from a recent review from the Ed Markut group, and summarizing the different kind of way that you can humanize yeast. And in addition to characterize, directly functionally characterize the disease alleles in yeast, mostly based on the concept of functional complementation, asking the question whether or not a specific disease alleles could replace the corresponding homolog. Sorry. And yeast is also a great model organism to detect genetic interactions. So in here, what we're trying to do is trying to develop an assay where that we can detect genetic relationships, suppressors or enhancers between human gene alleles in yeast models. And the reason, and then here, that in the next few slides that I want to show you a story that we're able to publish this a few months ago. It's also actually another postdoc project and continue until I have my own lab. And we asked the question is, to what extent that we think human genes could be functional in yeast? There had been a really nice paper published last year from Ed Markut's group, and they're systematically testing to what extent human ortholog can replace yeast, corresponding essential yeast homologs. And their conclusion is that it could be up to 50%. And a bit different from what was shown in that study. In this case, we're asking the question to what extent that human protein could physically interact with yeast proteins. And to do that, we carried out a systematically screen, a yeast-to-hybrid screen, of about 8,000 human protein coding genes with about 4,000 yeast protein coding genes. And here, the distinction is that we do not differentiate human genes whether or not they have a corresponding yeast homolog. We put them in the screen. And we made these choices for two reasons. Number one, Mark, my postdoc Winter, wants to find out to what extent proteins that are unrelated to each other can still physically interact. And for me, I always curious about to what extent that the functional homolog that potentially lacking sequence similarity could be detected in this kind of screens. And to summarize the story short, we come up with the interspecies network. That is hardly distinguishable from the intraspecies human-human and yeast network. And what we can expect before the experiments, obviously, where could we find interspecies interactions? There could be conserved proteins, conserved protein-protein interactions between human and yeast. And shown here is a pair of yeast protein, that's orange, interacting, and their corresponding human homolog are also interacting if the binding interface is conserved. And then we can definitely expect to find interspecies interactions between a protein and the corresponding homolog of its interactors. And following this hypothesis, we find indeed there is significant overlap of such cases in our data set. In such conserved interaction, likely underlying some of the known cases of functional complementation and corresponding to highly conserved domain-domain interactions between human and yeast. And when we're comparing the entire data set of human yeast across species with the yeast and human-human interactions, we found the overlap, meaning that human yeast and homolog share mutual interactors are significant across the entire data set, not only just for proteins that can complement. And most interestingly, we found that sometimes the shared mutual interactors are not necessary to be conserved between human and yeast themselves. What this suggests is basically that conserved ancestral binding mechanism preserved between human and yeast homologs could potentially mediate in species-specific interactions underlying species-specific functions. And we also find, consistent with this kind of a hypothesis, is that there are human non-homologs appears to interacting with highly conserved yeast machineries, and indicating that this might be a way of how complex the organism gaining new functions by having new interactions evolved from conserved binding interface. And based on this, And we know that coming back to our big problem is trying to identify genetic interactions between human gene alleles. We know that for everybody here that the yeast has been a system we can, characterize genetic screens, identify suppressors and enhancers. And in this case, we propose, we can start with the humanized yeast model and also doing interspecies identification of suppressors and enhancers. And there are a number of studies that have been done in, for example, humanized yeast model expressing human disease alleles, or in other cases that there are a number of human disease proteins that are associated with neurodegenerative diseases are overexpressed and they cause toxicity. And there have been yeast suppressor genes and enhancer genes identified across using the genome-wide deletion mutant strands or genome-wide overexpression strands, or even point mutation, spontaneous mutations. And so here we're proposing is that maybe we can also take one step further and to do genetic screens using overexpression human gene library to find out potential human suppressors and human enhancers of humanized yeast models. And to achieve that goal, basically in my own lab, we're able to construct a yeast-expression human gene library. We started with a full-length sequenced human gene Orpheum that contains about 15,000 human genes. And we did gateway cloning. We did cloned these human genes into yeast-expression vectors under the GAL1 promoter. And we put this library into a few genetic screens. And the idea is shown here that under either repressive or de-repressed conditions, we hope to find the human gene suppressors, shown here as red, that confers growth advantage, and human gene enhancers that confers disadvantage, that's shown here as blue. And basically, we put forward several different kind of screens that for humanized yeast models expressing disease-associated alleles and expressing neurodegenerative disease-associated overexpression of human genes, and trying to identify both suppressors and enhancers. And we think that such systematic platform can be used to quantify, whether or not there could be suppressor and enhancer relationships for allele-specific genetic interactions. And basically, we can ask, generate humanized yeast models that expressing different type of alleles. And most importantly, we think that this could be a platform where it can be complementary for understanding the cellular mechanism by following up with physical interaction screens and identify the yeast interactors of the human genes in yeast cells. Because we think there are only at least two possibilities of the suppressor or enhancer interactions that can be established in yeast cells in one way, that these two human gene alleles might physically interact. In the other, these two human gene alleles and potentially have mediator yeast proteins. So with that, I would like to thank my Department of Biological Sciences and the PhD program at Wright State University, and they have provided the people the funding to start my lab in the space. And this is the group of today. My first two PhD students just joined the lab, and collaboration with my husband's group on Shulinju, and he has a poster that also summarizing some of the screens that we are doing. And I would like to thank a funding from a research foundation that provided the funding for 2 years for us to able to do some of the screen, most importantly, generated the library. But the only request they ask is that they need to remain anonymous. And I also want to thank my PhD and postdoc mentors. And I would like to have a shout out of the SGD database, and which I fell in love with when I was a graduate student, and is still in love with it until today. It's just such a great resource for teaching and for get students started. And thank you all very much for your attention.
Is the library available, or will it be made available soon?
Oh, I'm very happy to hear that, and I thought that I need to take another couple of presentations before I can convince anybody to consider using a library as such to find the potential function for your interest. But right now, we have not yet made it public available, but immediately after we potentially publish our first paper, and we would very much like to do so. But we're happy to collaborate if there are people who are interested in even before that happens. Yeah.